It's a pleasure to introduce Zohar Sachs for today's Grand Rounds. She's currently a tenure-track assistant professor in the Department of Medicine in the Hematology division, and she's a recipient of the Lois and Richard King Assistant Professorship in Medicine. That's terrific. And she's a physician scientist, but with a clinical focus in hematological, I can never say the word, malignancies, but she also has a very active research program, and she's going to share some of that with us today. And her focus really is on defining vulnerabilities that impede successful treatment of vulnerabilities in the tumor. And her specific interest is in mechanisms of self-renewal, cellular self-renewal. Zohar, that's something you'll cover today, I assume?
Yes.
Oh, fantastic. She uses a combination of animal models, of stem cell assays, novel computational approaches in her studies. And particularly impressive, as I was just having a conversation before you all got online, her bachelor's degree was in chemical engineering, which that's absolutely, I think, amazing. So most recently, she's been using single-cell RNA-seq mass cytometry, or CyTOF, which is certainly a technology that's coming online into its own now to define these signatures. And I think she'll tell us a little bit about those today. And I think based on seeing a list of her co-authors and some of her papers, I'm sure a lot of you already know this about Zohar and her interest both clinically and her academic research interests. So I'm delighted that you are able to share your work with us today, Zohar, and I turn it over to you. Thank you so much.
Well, thank you. Thank you for that. Really very, very generous introduction. I appreciate it. And thank you for giving me this opportunity. So I'm going to share my screen, right?
Bill's in charge of that.
Okay, Bill, let me know if I'm sharing my screen, and I'm going to put it in presenter mode. Or try
to. It looks good on my end. I'll be quiet now. I'll mute myself.
Okay, and you guys have presenter mode. And, I know it's kind of awkward with the Zoom format, but if you want to interrupt me at some point, please, just go ahead. I would love to hear, if you have any comments along the way, just feel free. And also, I should say, for those of you who attended the Cancer Center seminar symposium last week, I did try to change this talk from that talk at least a little bit. So I hope not to bore you too much. If you were there last week, this will be a little bit different anyway. Okay, so yes, my name is Zohar. I study acute myeloid leukemia, AML, and specifically I'm interested in AML with P53 alteration. So I really like this slide, and I apologize if you've seen it before, but to me, this is what makes this work so exciting. That when I started my faculty position, especially for AML, we had nothing new. We were at this premier medical center taking care of patients, doing clinical trials, but there was really in the upfront setting, there was nothing new in AML for 3-4 decades. I was doing the same thing I would have done if I were here, in the early 80s. And that always bugged me, and then in the last 5 years or so, you see all this list of medications. These are all drugs that have been FDA approved for the treatment of AML in the last 5 years. So all of a sudden in the last 5 years, we see this explosion of new treatments coming online for AML. And these new treatments are all rationally designed, targeted therapies for the most part. And these are drugs that came to us because of the work that people are doing in the lab, the basic science, the basic biochemistry, genetics, molecular biology. So the molecular revolution in biology has been going on for many decades now, but right now I'm really feeling it as a physician that we are really reaping the fruits of that work. And AML patients are living longer. They have more options. That first relapse is not an immediate death sentence anymore. And that's just new in the last few years. So I find that super, super exciting. Unfortunately, even though I have a pretty nice list here of drugs I can use that will work for various types of AML, none of these drugs work for AML with P53 mutations. So P53 mutant AML is treatment refractory, and none of these targeted agents work in that setting. Okay. So I just want to remind us of the way AML patients are treated. Basically, we have the standard of care chemotherapy. It was established in the, late 70s, early 80s. And the reason why we still use it for almost every patient that comes in is that it works. Most patients can achieve a complete remission. Unfortunately, despite this early response, most patients will relapse, and relapse eventually, leads to death in most patients. So most patients with AML end up dying of relapse disease, which is more of a problem than that upfront disease. And so then if you think about, well, if the real problem is relapse, what's causing relapse? And we think that there's a subset of cells that can cause relapse, and we call those leukemia stem cells. Okay, so we focus on leukemia stem cells. Leukemia stem cells are defined functionally by the ability to recapitulate the leukemia, which is kind of conceptually tricky, I think, to think about, because most of the leukemia cells are rapidly proliferating, but they have kind of a limited lifespan. They can proliferate a few times and then they die. Leukemia, as I guess this audience knows very well, is not just one kind of cell. It's a heterogeneous population of cells, just like any tissue, really. And there's a subset of those cells that can recapitulate that full spectrum of leukemia subtypes, cell subtypes. And the ability to do that is called self-renewal. We usually identify leukemia stem cells by stem cell markers. But, in targeting leukemia stem cells in AML therapeutically has been really elusive in AML. We haven't really managed that. And really the stem cell markers, those CD markers that we use to identify stem cells, they identify a subpopulation of cells with stem cell capacity or enriched with stem cell capacity, but they're not really good markers of function. So like you would think about CD34 on a leukemia stem cell, it doesn't necessarily reflect a self-renewal function. It's just a marker, we think, or at least we don't actually really know. And so I'm going to just tell you a little bit about the work we've done in the past and how that led us to P53 mutant AML. So we got really interested in this concept that leukemia stem cells are identified by these markers, but these markers don't reflect functional capacity. And we thought, well, if you really want to understand what's driving self-renewal, you need to understand the molecular mechanisms of self-renewal. And we thought, what's a better way to understand molecular mechanisms than gene expression profiling? But it became very clear that even within this and let me see if you don't see my pointer, but even within this leukemia stem cell population, not every one of these cells actually is a stem cell. We call it the leukemia stem cell because it has the markers of stem cells, but not every one of these cells has self-renewal capacity. So to understand mechanisms of self-renewal capacity in a very precise manner, we took these stem cells from a mouse model and performed single cell RNA sequencing. And here's a heat map of the gene expression profile. And right away, you see that there's heterogeneity at the single cell level, where there's a group of cells that seem to express these genes, a group that express those genes, et cetera. And so I'm just going to summarize this data. We published it a couple of years ago now, but basically we asked whether these different gene expression profiles that we see within the stem cell compartment, whether they correlate with differences in self-renewal capacity. And so that experiment was done by identifying cell surface markers that correlate with these gene expression profiles, sorting according to leukemia stem cell markers and according to the genes, that are differentially expressed here that are cell surface markers. And then sorting those cells and transplanting them into mice. And we found that cells that correspond to this gene expression profile were actually rapidly proliferating, but they were unable to recapitulate leukemia in a mouse. Whereas these cells with this gene expression profile and this CD marker expression, these were actually very poorly proliferative cells. They didn't proliferate a lot, but these were the only group of cells that could really efficiently recapitulate leukemia in a mouse. And so that whole project taught us really that there is a subpopulation of cells within the stem cell compartment that self-renews, and that we could identify that population based on its functional characteristics, its gene expression profiles. And then I just want to point out that the gene expression profile that corresponds to self-renewal in this case was full of NF-kappa B target genes. And in fact, the CD marker, CD69, is an NF-kappa B target gene. So it got us thinking a lot about the role of NF-kappa B in self-renewal in AML. And we're not the first people who noticed that NF-kappa B seems to be important for AML and for self-renewal, but we noticed it very, particularly at this single cell level in this project. Okay, so as we were finishing up that project, this was a really exciting project. It was a nice mouse model. We actually, as part of that previous project, we did study some primary patient material and saw the same thing in patient samples. So we were pretty excited about, focusing on leukemia stem cells. But meanwhile, clinically, I think all of you would know we're starting to see a lot of p53 mutant AML cases. And the reason we're seeing them is simply because the NGS lab is now doing NGS for us. So we recognize when someone has a p53 mutation. And so I started reviewing that literature more carefully, simply because I had patients with this disease. And like I told you before, when we treat AML, most AML goes into remission. And that's actually true for p53 mutant AML as well. We can get p53 mutant AML to go into remission or like a really, close to a remission where you're seeing a nice drop in blasts. Maybe you're still seeing some residual p53 mutations, but it looks pretty much like a good morphological remission in most patients. But these patients always relapse. Like I couldn't even get them to transplant. I could barely get them, through a few weeks or months after their induction therapy. And so I started looking at all these drugs that were coming out, and I thought, well, have any of these been tried in p53 mutant AML? And over again, if you do a search for p53 mutant and AML, what you find are all these papers that say that this drug works based on p53 function, meaning you have to have intact p53, or it doesn't work in p53 mutant AML. And basically, that's where I got kind of really more interested in this disease, right? So, and sorry, I guess this slide kind of tells you what you guys already know very well, that in AML in general, mutational profiling predicts, survival in AML, and that in a p53 mutation in AML is really invariably fatal. And that's what we're looking at here is survival curves of p53 mutant AML versus other types of AML.
Zohar, can I ask a question?
Yeah, absolutely.
Well, so maybe the rest of the faculty knew this well. I don't. P53 mutant, p53 is this black box for me. It's a mutant gene. What is mutant? Does it not work? Or does it have some sort of altered target? Or what does that mean exactly?
That is such a beautiful question, I'll tell you, because I think the field is very conflicted on what that means. And so that's a really, and I'll have to tell you that when I first took an interest in p53 mutant AML, I naively assumed that mutation means loss of function. And I think that the whole field sees it that way. And if you look at the literature, people often interchangeably use the word p53 mutant and p53 deleted. Right. So they'll use p53 double knockout cell lines and mouse models as as models of p53 mutations. So most people think that, and most p53 mutations hit in the So p53 is a transcription factor, right? And most of the mutations hit in the DNA binding domain, and basically you get loss of binding to p53 target sites. Okay. So that all is like very nicely identifying a loss of function, right? However, especially in solid tumors, and this is where it becomes really interesting for me, right, as a hematologist, and I only go to ASH and don't pay attention in the solid tumor world too much. The solid tumor world knew this way before we knew it in leukemia, which is this, that if you do look at p53 mutations, not deletions, so for example, I think the first evidence of this was a long time ago, maybe 10, 15 years ago. They showed that a p53 mutant mouse has a different scope of a broader scope of tumors than a p53 deleted mouse.
Okay.
And a p53 mutant mouse is more likely or has more metastatic lesions than a p53 null mouse. Okay. So that was, I think, maybe the first hint that the p53 mutations are actually providing something beyond loss of function.
Okay.
And more recently, it's been shown in AML as well that a p53 mutation provides something to the cell that is not just loss of function. And I've seen some really interesting data at AACR, for example, that suggests that that mutant p53 proteins bind transcriptional regulators, like different transcriptional regulators than a wild type p53.
Okay.
Sure. And then a few people in AML now, and that was like in a sort of, I don't remember, some cell model that wasn't in leukemia, but in leukemia and in hematopoiesis, people have shown that mutant p53 bearing stem cells have enhanced self-renewal capacity and have an alternate gene expression profile. So it's not just that you lose p53, wild type p53 driven target genes, but you have these other genes that are expressed, and you have a pattern of binding to the genome that is different than wild type p53. So I personally believe strongly in this data. I think it's really very convincing that a mutant p53 is not the same as a knockout p53, and that it is doing something. And I guess I'll like one more thing I'll say about that is in AML specifically, p53 mutations aren't super common. They're fairly rare, but among mutant p53 patients, they very rarely lose both copies. They very commonly lose one copy. So you see loss of heterozygosity in like 70% of cases. That loss of heterozygosity, not loss of heterozygosity, they call it biallelic targeting, because you could have a mutation and a loss, or you can have two mutations. So like 70% of cases will have that, but very, very few, less than 10% of those will be biallelic loss. So AML almost always chooses to keep a mutant protein around. Okay. Anyway, so this is sorry to.
No, that's very helpful. Thank you.
And I find it super interesting. Yeah, but it's
Zohar, this is Chris Pennell. I just have one follow-up question.
Yeah.
Are the leukemic stem cells in refractory disease refractory because they've lost expression of the target of the small molecule inhibitor?
That's a really good question. And oftentimes it's complex, right? So oftentimes it's not just loss of expression. Like, so for example, a FLT3 mutant AML, like the most common ways that FLT3 mutant AMLs become resistant is expression of NRAS, a mutant NRAS, developing NRAS mutations. So they basically, if you have a FLT3 mutation, you rely on it, we inhibit FLT3 activation, and then some other, NRAS sort of bypasses that. So that's one common way. So let's see, so the IDH1 and 2 mutations, I'm not actually sure. I don't remember exactly what if I know their mechanisms of resistance.
That's fine. So it's just complex. Okay.
It's complex. I would definitely say it's complex.
Right,
and I would say also, AML we think a lot of those mutations are subclonal. And so you just get some other clone growing out oftentimes. Yeah. We think p53 mutations become clonal as you treat with chemotherapy, and if you had a subclonal p53 mutation after chemotherapy, it'll become more dominant. Okay, so clearly, because I guess let me just make that point clear that when we treat p53 mutant AML, it does either achieve remission or have a really good disease control in most cases, but it always comes back, meaning basically that p53 mutant AML, like other AMLs, is a leukemia stem cell driven disease, right? But unlike other AMLs that are curable with either chemotherapy or targeted agents or transplant, p53 mutant AML is never curable. Even if you get them to transplant, they relapse. And so we think really there is something unique about those stem cells, those p53 mutant stem cells. And so that's what brought it back to us, to our lab, where we think, well, we know how to deal with stem cells. We know how to study stem cells. And so can we learn something about the molecular features of p53 mutant stem cells that are critical for self-renewal that we could target, really? Okay. So really, I wanted to just highlight the central concepts here, right? Which is that self-renewal is a feature of only a subset of cells, and that we could, based on our earlier work, really model that understand that by a molecular state, right? There's a specific molecular state that drives self-renewal, and that we think p53 mutant stem cells are uniquely treatment refractory. So the first kind of easy question to answer is, do mutant leukemia stem cell, p53 mutant leukemia stem cells harbor a unique molecular state? And even though a lot of our previous work was with gene expression profiling, we thought to kind of jump the gun to ask a more translational question, and that took us to signaling pathways. So we know that in AML, signal transduction pathways are hyperactivated in almost every disease. And there's data to show that they are hyperactivated in p53 mutant AML as well, just like any other AML. And the other thing about signaling pathways is that we have drugs that inhibit signaling pathways, right? And I'm guessing you guys will, recognize some of these, like RAF, MEK, mTOR. These are ones with, pretty famous BCR-ABL, JAK, pretty famous inhibitors to those drugs, to those signals. And the drugs that inhibit these signals are, really pretty effective in certain settings. Anyway, so we decided to just profile the signaling pathway activation state of stem cells with p53 mutations. And so we decided to do that with CyTOF. So CyTOF is like flow cytometry, basically, where you get quantitative measurements of proteins at the single cell level. And with flow cytometry, you could measure, 7, 8, 10, 12 different proteins in one cell, at the same time simultaneously. So CyTOF allows us to do that on a slightly larger scale. So we can do up to like 40 proteins simultaneously at the single cell level and quantitatively. And the reason why we turn to CyTOF and why we want all those channels is that we want to be able to identify stem cells, right? So we use up a bunch of markers just looking at our stem cells. So a bunch of channels are used up there. And then we wanted to look at a bunch of signaling molecules all at once, so we can ask a question about how these signaling molecules interact with each other. So that's why we went to CyTOF. And here we're looking at it. It's a pretty complex figure, and months and months worth of work all in one figure. But basically what we're looking at is we took primary human AML samples and we grouped them according to their genetics, right? So normal, kind of intermediate risk AMLs, FLT3 mutant, AMLs, NRAS mutant, and MLL rearranged AMLs, and then these poor risk AMLs that are p53 mutant. And we just measured within their stem cells all of these different markers. And Ki67, a lot of you will recognize, is a proliferation marker. And we know in the stem cell field that stem cells are poorly proliferative, right? And so the fact that the leukemia stem cells of most of these AML subtypes were poorly proliferative is what we expected to see, and we were happy to see that what we expect to see is there. Okay, but now we go along and look at all these different signaling molecules and ask, is there something unique about the p53 mutant stem cells? And what we saw is that NF-kappa B seems to be upregulated in these stem cells, and 4EBP1 seems to be, I would say, uniquely upregulated in p53 mutant stem cells. And these are just primary patient samples that we profiled. So based on this, we thought, well, maybe there is something unique about these stem cells, and maybe they depend on our old friend NF-kappa B, and we also noticed this 4EBP1. Okay, so we tucked that away and then turned our attention to testing a bunch of drugs. Basically, around that time, there was a paper that showed that in P53 deleted. So now if you don't have P53 AML cell lines, there were certain drugs that seemed to work better than others. So we kind of started with those drugs, and we gathered a bunch of drugs from the literature that for one reason or another thought might work in P53 mutant AML. And we thought, why don't we just profile these? So for these experiments, we took primary human AML samples with P53 mutations and treated them with these drugs in vitro. And then we did a bunch of assays. And the first assay is a pretty simple one where we just measure leukemia stem cell frequency in the drug treated over vehicle treated for each sample. So each, circle here represents a different leukemia sample. And I put a little star next to these drugs that seem to reduce leukemia stem cell frequency. And some of these drugs didn't. So I just put one example here. What did I want to say? Oh, this was a really important assay, the leukemia stem cell frequency assay, because we know that even though P53 mutant leukemia responds to chemotherapy, meaning the patient achieves remission or the blast percentage goes down, we know that when you look at, like in mice or in vitro, even though you see loss of leukemia cells, like the percentage of the leukemia cells die, the percentage of cells that remain alive, the percentage of stem cells that remain alive is high. So basically the chemotherapy spares stem cells, and we see an increase in stem cell frequency after treatment. So we thought we don't want to be fooled by things that just kill off cells, right? And I would say that the screens that identified these drugs didn't look at stem cell; they just looked at cell numbers. So we thought, we can kill off leukemia cells. We know how to do that. We have good chemotherapy for that. What we don't know how to do is to kill off stem cells. So that's why this was a really important assay for us. But again, here we're just using stem cell markers. So we wanted to go to a functional assay, and for functional assays, we use colony forming assays. And technically speaking, to really look at self-renewal, you need to do serial colony forming assays. So you plate them, every colony that forms, is originated from a single cell. You pick that colony and you plate it again in colony forming assays. And so that capacity to form serial colonies is the surrogate for self-renewal capacity. But what you'll see here is that we had a couple of these drugs that abolished all colony formation. And that was true even when we used them at super, super low doses. So we were really unable to do serial colony forming assays with these drugs because they just abolished all colony forming capacity. And I just, draw your attention to venetoclax that did not lower stem cell frequency in these assays, and also had some effect in the colony forming assay, but not as much as some of these other drugs. And venetoclax is really, frontline therapy for elderly patients with AML. So that's why we like to use it as a control. Also, venetoclax has been shown not to work in P53 mutant AML. Anyway, okay, so well, let me just summarize here that we think we found a few drugs that seem to work. They reduce stem cell frequency, and they seem to abolish stem cell capacity in the best way we have to measure it. So we went back to the CyTOF and did another CyTOF experiment where we just compared the expression of all those different signaling molecules that I showed you before in the stem cells of a bunch of P53 mutant AML samples. And here I'm showing you just one signaling intermediate, because we found that these drugs that worked in our stem cell assays reduced NF-kappa B. And here we're looking at Visny plots. So in this CyTOF experiment, the way we analyze it is this Visny analysis, which basically places every cell as a single dot on this plot. And then the color of the plot represents the amount of whatever intermediate we're looking at. So here we're looking at NF-kappa B. We could look at a bunch of other things, but I'm just showing you one representative sample just to show you that crizotinib, which is a ALK inhibitor, and AZD, which is a JAK inhibitor, reduce NF-kappa B levels really very substantially in a drug-dependent manner. And venetoclax, which I just mentioned is it's a BCL2 inhibitor. It works really well in frontline AML, but does not work in P53 mutant AML. Venetoclax seems to increase NF-kappa B rather than decrease it. And this is just a representative sample, but we saw that in, multiple samples for actually multiple drugs. So the drugs that seemed to target stem cells in our stem cell assays, all of them reduced NF-kappa B levels. And the drugs that didn't work did not reduce NF-kappa B levels. And then we talked about 4-ABP1. And here I'm showing you, again, another some more CyTOF data, but here we're looking at 2 or 3 different primary human AML samples with P53 mutations, and we're looking at 4-ABP1 levels after treatment with all these drugs. And these 4 drugs here that lower 4-ABP1 levels, these 4 drugs were all drugs that lowered NF-kappa B levels and also reduced stem cell frequency and colony forming capacity in our kind of more functional assays. And that, remember that GW drug that we talked about, it's a CSF1R inhibitor. It does not lower 4-ABP1 levels, or maybe it even increases them. Okay. So then, oh yeah, sorry. So let me just conclude here that what we saw is that we found a bunch of drugs that seem to work. These different drugs that seem to work in reducing stem cell capacity, stem cell frequency, all of those drugs lowered NF-kappa B levels, and they also lowered 4-ABP1 levels, even though they all had different kind of predicted targets, right? I told you, like, an ALK inhibitor, JAK inhibitor, et cetera, but these guys, what they had in common was NF-kappa B and 4-ABP1. Okay, so we sort of got interested in this concept and thought, well, is it that NF-kappa B is driving self-renewal in P53 mutant AML? So we wanted to ask that question in kind of a different way. And remember, I told you in the very beginning of the talk that we published that CD69 marks a self-renewing subpopulation of mouse leukemia cells, and that CD69 is an NF-kappa B target gene. So we thought, well, could CD69 be marking a subset of P53 mutant leukemia cells with self-renewal capacity? And so we sorted these primary human AML samples based on CD69 expression, and basically the CD69 high fractions formed many more colonies than the CD69 low colonies fractions. And you'll notice that for some samples, those CD69 low fractions didn't form any colonies. So we couldn't even perform secondary colony forming assays with these because there were no colonies. But with some of these guys, we could. So we picked the colonies from both of these fractions, right? And we didn't sort again. We just took what grew here and what grew here and plated them in secondary colony forming assays. And what grew here was unable to form colonies again. And what grew here formed colonies again. And here it's just number of colonies. So this is like 25. It's not even by the axis, it looks close to 0, but it's a lot of colonies. Anyhow, so basically this data showed us that CD69 marks a self-renewing subset of P53 mutant AML, which makes us think that maybe, again, all roads are leading us here to NF-kappa B, right? And so we compared NF-kappa B levels actually in the CD69 high compartment of P53 mutant AML and intermediate risk AML. And we actually see that P53 mutant AML with the CD69 fraction of those samples actually particularly upregulate NF-kappa B relative to other AMLs. Okay, so all of that put together led us to formulate this hypothesis, right? So we saw a lot of evidence that it seems like NF-kappa B and 4-ABP1 were elevated in P53 mutant stem cells, and drugs that can lower those intermediates seem to reduce colony formation, reduce stem cell capacity and stem cell frequency. So we started to think that maybe P53 mutant AMLs are dependent on NF-kappa B and dependent on 4-ABP1. Okay, and if you do a search in the literature to put those two signaling proteins together, you won't find anything or hardly anything. But I want to just review with you why we think they're probably linked. Okay, so 4-ABP1 4-ABP1 gets phosphorylated in response to mTOR activation. So mTOR activation leads to elevations in phospho 4-ABP1 levels. And when phospho 4-ABP1 levels goes up, you get an increase of translation of oncogenic proteins. And this axis is just, very, very well documented to be a really important oncogenic pathway. And mTOR is widely credited for oncogenic protein production, right? It sort of revs up the cells to make tons of proteins. And that function of mTOR is through phosphorylation of 4-ABP1, which allows for translation essentially. Okay, so this kind of axis is very, very well known. However, what's also known is that when you make a lot of proteins, sorry, I just want to see the time. Okay, yeah, we're good on time. When you make a ton of proteins, when the cell is making a ton of proteins, it makes a lot of mistakes. So it makes a lot of defective proteins. And we know that accumulation of defective proteins is toxic. So this unfolded protein response will get activated and you get cell toxicity. So we know that in its oncogenic effects, mTOR also has this other kind of byproduct. And in BMBB at the University of Minnesota, Do-Young Kim is a faculty member there, and he really has done a lot of this seminal work in mTOR biology, I would say. And what he discovered a few years ago now is that in order to tolerate this excess defective protein production from mTOR, mTOR also upregulates proteosome function, and it does so by the assembly of the immunoproteosome. So not the constitutive proteosome, but the immunoproteosome, which is an inducible proteosome. So mTOR causes assembly of the inducible proteosome. This is a post-translational modification. It's not at the gene expression level, it's at the protein level. And this increase in proteosomal capacity allows cells to survive this protein stress because basically they can, they can get rid of these defective proteins. So we collaborated with Do-Young Kim on that paper simply to show that this axis is active in AML. So AML with hyperactivated mTOR has increased assembly of the immunoproteasome, and that this is important for colony formation in AML. Okay, so we showed that. And so now when our data in P53 mutant AML is pointing to 4-ABP1, we think about this axis. And then the other thing that people know, lots of people have shown, is that, right, that NF-kappa B is important for AML and AML self-renewal, we know that. But what we, a lot of people know very well is that the proteosome degrades an inhibitor of NF-kappa B, right, I-kappa B. And so excess proteosome function will lead to upregulation of NF-kappa B function. And we know, for example, in multiple myeloma, that when we inhibit the proteosome, we inhibit NF-kappa B. So the proteosome inhibitors are widely recognized as NF-kappa B inhibitors, okay? So sort of this piece is well known and this piece is well known, but nobody's really put these pieces together that elevated 4-ABP1 might be related to elevated NF-kappa B, and more importantly, malignancies with with high levels of phospho 4-ABP1 are dependent on the proteasome, and NF-kappa B is dependent on the proteasome. And so if you inhibit proteosome function, you will reduce NF-kappa B, but you will also potentially kill malignancies that are dependent on phospho 4-ABP1. So this is like a really long-winded way of saying we wondered whether this whole axis was related in P53 mutant AML. And one other thing I would point out is that mutant P53 upregulates NF-kappa B. That's been shown. How it does that, nobody knows, but we know that when you induce mutant P53, you get induction of NF-kappa B. Malignancies with mutant P53 have hyper NF-kappa B. That's been described not in AML, but in other solid tumors. And in embryology, not in cancer, mostly in embryology, mutant P53 was shown to upregulate mTOR. And wild type P53 is known to attenuate mTOR and NF-kappa B. So there is some data tying P53 and mutant P53 into this whole axis. And so this is sort of, the prelim data led us to sort of put together this model. And so we asked a very simple question. what is the impact of proteasome inhibition on p53 mutant stem cells? And we predicted that this could be a really effective way to target stem cells in p53 mutant AML. And so here I'm just showing you a Visney plot to show you that within the stem cells, you see NF-kappa B is like nice and brightly red. When we treat with this immunoproteasome inhibitor, so this is specific just for the immunoproteasome, we see loss of NF-kappa B. And here we have quantitated that vehicle, and then here we're using carfilzomib or PR957, and we see a loss of NF-kappa B. And here, and these are all in primary human AML samples looking at the stem cells specifically. And here we're looking at 3 different proteasome inhibitors. So bortezomib and carfilzomib are pan-proteasome inhibitors. They'll inhibit the regular proteasome, the constitutive proteasome, and the immunoproteasome, whereas this is a PR is a 957 is a immunoproteasome specific inhibitor. Anyway, they all lower NF-kappa B levels in the stem cells of primary human AML samples. And this was, each of these were statistically significant. Okay. So then we asked the functional question. We treated primary human AML samples with proteasome inhibitors and looked for colony formation. And here, for example, carfilzomib and PR957. And this, look at this axis. So one is up here, right? So if it has no effect, it would be none. But both of these drugs reduced colony formation relative to vehicle. And then when we took these samples we didn't treat them again, we just plated them in secondary colony formation assays. And you see vehicle treated samples can form colonies, but so samples that were treated with proteasome inhibitors way back in the beginning of the experiment cannot form colonies. So this is kind of the classic assay for self-renewal, and we see very nicely that proteasome inhibition abolishes self-renewal capacity in primary human AML samples with p53 mutations. And so it really lends a lot of credence to this model, at least this part of it. I'm not sure whether this arrow belongs here. And I wonder whether the whole thing just goes through mTOR, whether p53 mutations elevate mTOR, which elevates immunoproteasome, which leads to elevated NF-kappa B, or if there's another way by which p53 mutations upregulate NF-kappa B. We don't know that, and we are working hard to figure that out. We actually have a genetically engineered mouse model of AML where we're going to try to figure this out, really with some strict genetics using a, well-defined model. But we also have primary AML samples and PDX models where we're working on this. And then we are, I didn't mention any of these, but we have some people working on these more single cell RNA-seq data analysis and some developing methods for network analysis in CyTOF data, et cetera. So the data I showed you was generated almost entirely by Clara and Marie in the lab. Yun-Kyu and Daniel are graduate students who also do a lot of really exciting work I didn't show mostly. This woman goes by June, and she's working on, some of you may have heard there's a p53 stabilizing drug. So we're checking that drug. Doesn't seem to stabilize p53 in AML, but we'll see. And Kirthana and Sam are students in the lab. All of the samples we got from the heme malignancy tissue bank, obviously the mass cytometry, the CyTOF course facility was instrumental. I work very closely with David Largaspada, who contributed a lot to the work I showed here. Chad Myers and Karen Sachs also, and Mike Linden actually contributes quite a lot to everything we do. He does a lot of pathology analysis for us. And anyway, so, and of course, I thank our sponsors and my division, and thank you very much for listening. And if you have any questions, I'd be happy to review them.
Zohar, thank you so much. That was terrific. Really very well done. There's a question on chat for you to start from Dr. I hope I can pronounce her name, Gatak. Maybe Dr. Gatak could actually ask the question. And there are 2 questions now based on.
Yeah. Okay. So I see this. So, okay. Is it based on loss of function? So the question, the first one I'm reading here is NF-kappa B, is that a loss? Is that due to P53 loss of function or an aberrant gain of function? That's a really good question. You know, like we discussed earlier, we don't know. We didn't test that. And I love that you asked that question because we are going to test that very directly. So our next set of experiments are to knock out mutant P53 and not overexpress mutant P53 and measure NF-kappa B. So that's a really good question. We just don't know. There's some data, I guess, in gene expression from other people that help us, but I think we just have to do the genetic experiments, and that's what we're working on. We're just testing for NF-kappa B right now by measuring it by CyTOF. Of course, we want to do gene expression analysis and all that stuff. So we did do gene expression analysis in that single cell gene expression data set. We haven't done any gene expression analysis of P53 mutant samples yet, but we are, kind of eager to do that, as soon as we can afford it, basically. And we don't really know what's going on with, we don't, we presume it's through the proteosome. You're asking about I-kappa B degradation and DNA binding. We haven't done any of those kinds of assays, but those would be really interesting.
I have a follow-up question to P53 again. And so there are different mutations of P53. When you talk about a P53 mutant population, are they stratified, subdivided for different types of mutations, or is it just all in one big grab bag of mutations?
That's a really, really, really good question. So right now we just have, I think every sample for which we know the mutation, it's always a DNA binding domain missense mutation. We have a few samples for which we actually don't even know the mutation. We just know that there is a mutation based on the cytogenetics, like loss of 17p or something like that. Okay. We have all of those samples. They are being, in fact, they've been sequenced. I'm just waiting for the data analysis. But that's why we brought in, maybe I didn't mention it very clearly, we brought in a mouse model so that we can test it like in a mouse model that has like one single P53 mutation. Sure. And then we're going to do all those like lentiviral P53 knockout assays in our primary human samples.
Absolutely.
I think it would be really beyond the scope of what we can do to start classifying mutations too much, because there's frame shift, there's like, truncation mutations. But I think in general, what I would love, like my goal is that hopefully we will find something that might be true for like most of the mutations.
Got it. Okay, thank you. Are there other questions for Dr. Sachs?
Hey, Dr. Sachs, can you hear me? I have a quick question.
Yeah.
So one of the things that has been shown when for treating leukemic stem cells has been like inhibition of NF-kappa B as well as induction of reactive oxygen species. So do these TP53 mutant LSCs have any difference in the reactive oxygen species expression when they are treated or before treatment, or have you noticed any difference in them compared to the non-53 mutant AML LSTs?
That's a really terrific question, and I really appreciate that you asked. So we do notice differences in ROS. We've done a few measurements. We wrote a few grants. This drug right here, elescalomol, induces reactive oxygen species, and you see how very, very effective elescalomol is in these assays at very, very low doses. So, and P53 does. Really, it has, P53 is known to be part of the protective, redox response of the cell. So we do think that P53 mutant samples might be uniquely sensitive to like induction of extra ROS beyond what they have. So we've basically written some grants on this topic, and none of them have been awarded. So we haven't like actively pursued it because we've gotten some pilot funding to study some of these other, like the model I showed you. I find that question to be incredibly important. And in fact, what I didn't kind of mention, but I told you that June, one of the students is working on that drug that seems to stabilize P53, mutant P53. In our hands, we're not seeing evidence of induction of P53 in AML, but that drug also works by inducing reactive oxygen species. And so I wonder if the efficacy of that drug is really not because it's, making P53 normal, but because it induces reactive oxygen species in a cell that cannot tolerate it. But we don't know yet. We're looking into it. We're doing all the assays, like we're, doing, treating cells with that drug plus N-acetylcysteine and that kind of thing. So we don't know, but it's a really great question. Vanessa Dayton asked if we should be doing IHC for CD69 for P53, yes. Yes, IHC for P53 on all new AMLs would be a definite yes. For CD69, I would love to see that, but I have to admit that my data is a little premature, so we do need to do a little bit more work to validate CD69, but P53 for sure. Also for CD69, we're not sure what to do about it. CD69 is expressed on T cells, and it's not clear that it's a great target, even though it's a great marker.
But at least we have the data.
That's right. That's right. Yeah. I would love to have that data. But definitely P53 IHC. I think that would be very key going forward, and that would be clinically relevant and very important for our studies.
Which is now it is now considered routine in hematopathology. Good. To order P53 on new AMLs and or high grade MDS. But if we looked into starting routinely doing CD69 along with the P53, that would be additional data for you, would it not?
It would, absolutely, yes. Yes.
So that was my question. Thank you.
Yeah, thank you. Yes, that would be, I would love that. Yeah.
Very good. And Dr. Pinnell has a question as well. Thank you.
So Dr. Pinnell says, can you reduce LSC colony forming capacity by upregulating components of the immunoproteosome with, for example, IF and gamma? So we would predict that upregulating the immunoproteosome would enhance stem cell capacity, right? Because upregulating the immunoproteasome would increase NF-kappa B, and it would like allow cells to tolerate high phospho-4-ABP1. We haven't tried that experiment. I think that's a really good idea. What somebody suggested, kind of relating to that CD69 question, is somebody suggested treating them with a CD69 ligand. But yeah, on that interferon gamma question, definitely the kind of inflammatory microenvironment is thought to be very pathogenic and contributing to progression, at least in MDS, if not AML. Anyway, so that's a kind of a really intriguing connection there, absolutely, but we haven't tried that yet.
Okay. Do we have any other questions from the faculty? Zohar, absolutely terrific job. Thank you so much. And best of luck with future studies. It sounds like you have a full plate of studies to do.
Yes, yeah, we have a very full plate, but thank you so much. Thanks for the questions and the discussion. I really appreciate it.
Absolutely, our pleasure. Thank you.